Good afternoon, everyone. My name is Pattanai Konpetch. I'm a master student from the Faculty of Science, Mahidol University, Thailand, and today I'm delighted to present my own research project under the title of identifications and characterizations of phage depolymerases input from the Clostridioides difficile phage. Right. In Thai cultures, is the sayings, one has phage, is two has this, only three are survive. And this quote is quite similar to two has better than one in the Western world. Right. And this concept just not only fit in just only a human being, but it also fit in, even though it's just only a tiny single cell organisms such as bacteria. Right. Bacterial cell by itself also have ability to sense their own friends. That's how much of they are around by sensing the level of the specific messages in the form of the chemical compound, right? Then it seems to be popular density dependent madness. That means that the more of the cell they are, the more of the chemical releases from individual cell, right? And this mechanism is called apparent resistance mechanism. That bacterial cell can send a difference of their specific messages and decide what is the pathway that they want to upregulate it or downregulate it to respond to a changed environments and helping them survive from change of environments also. Right. And one of the pathways that we control under the apparent resistance mechanisms is about information. The part of them is a community of bacterial cells living together by enzymes themselves with their own secreted extracellular polymeric substance matrix or the EPS matrix that helping to attach to surfaces. And this matrix just not only provide adherence functions, but also act as a physical barrier to block out positive environments. For example, the host immune system, oxidative stresses, and especially the antibiotic treatment to reach into the cell under the buffering protections. Right, so letting out often exceptions of the antibiotics, which makes the antibiotics treatment that have long been a form of therapy against the bacterial infections might not effectively work anymore when you're fighting with the biofilm-forming bacteria, right? And as many of you guys here want to apply a phage therapy to fight against the biofilm-forming bacteria, that's called of a antimicrobial resistance also, right? And one of the advantages, that's why we thought that the phage therapy might be fit in to solve this problem, is might be that some of the phages, they're proposed to contain an enzyme that we call phage depolymerases enzyme. The enzyme that can catalyze both degrading, destabilizing, penetrating to make the biofilm more weaker, looser, and easier to get inside to the cell under the biofilm protections. And as a group of research, we focusing on identify this enzyme from Clostridioides difficile phage. Because of the Clostridioides difficile is about biofilm species, and also is concerned as an urgent threat to the human society following from the CDC report, right? And based on our previous study in our lab, that we also try to isolate and characterize phages from the clinical difficile isolates received from Thai patients from a Thai hospital. And we successfully received 5 different isolates of the phages in also one phage particle. Right, and this thing from all the candidates, the phage by Asian team here, it shows a potential to have the greatest infectivity and also a widest host range spectrum from our collections. But it's just not only the infectivity and the broad range spectrum of these isolates, but this phage also exhibits a resilience in using in a wide range of the temperature and pH also. And we also try to treat the phage isolates into the biofilm populations of the difficile, and the result turns that it's also effectively decreasing the variability of the cell, even though they are in the biofilm protections. And this is why this is where my research questions and so hypotheses were formed. That is, when you're looking back into the principle of the phage life cycle, right? The vital step that phage must to be success is must to be reached into the receptor that rely on the positive surfaces. And the existence of that matrix might be blocked them to reach into the receptor. So we thought that phage might naturally have some mechanisms to to make it more easier to get into the cell under the biofilm protections. And we thought that the missing pieces might be a phage depolymerases enzyme. So we started the in situ analysis to analyze for the coding sequences that might be coding for phage depolymerases inside the genome. And we use in two major criteria. For the first one is we're trying to screen any possible genes that can catalyze for depolymerization reactions also. And next, we also looking into locations because from several studies, they suggest that a hotspot to finding for phage depolymerase encoding sequences might be located at the attachment and lysosomes or nearby these organizations. Because of the attachment and lysosome, right, they responsible for encoding for the structure of the phages that involve in infecting and lysing of the cell wall. It might be the first part that started interactions with the host cells also to start the infection. So we thought that this might be these locations. And following from these criteria, we received three possible genes that might encoding for phage depolymerases. But standing out from all the candidates, the endolysins, which previously described as a cell hydrolysis enzyme, shows the effectiveness to be reproposed as a phage depolymerases enzyme. So, right. And if you're looking into the architecture of the endolysins with the cell hydrolysis that lies the bacterial cell wall from the inner part to the outer part of the cell during the lysogenic release, right? So if you look into the architecture, especially that's input from the phage part and chain, they contain of two major domains. For the first one, at the N-terminus is enzyme active domain or EAD, responsible for cell degradations, and another domains at the C-terminus is CBD or the cell binding domain, responsible for the specificity of the enzyme also, right? But in the case of the endolysin, right, they have a variety type of the EAD, depends on which targets that they target on. But for the C-different-L, they target on the linking between the N-acetylmolaminic acids with the L-alanine in the peptide step, right? And alongside with our phage study, we also perform a characterizations of the biofilm forming abilities of the 63 clinical difficile isolates and along with the 22 standard reprotype stain of the difficile also. And we found that we have a different character of the biofilm formations. For example, in the term of the morphologies, we received different morphologies of the biofilm formations. For the first one, we call the rooting biofilm, which they form and branching like the root of a tree. The second morphology is the centric morphologies that is formed dense at the center of the wellplets. And the final one is the edges biofilm that tend to form dense at the edge of the wellplets also. Right, just not only the morphologies, we also try to tracking on the quantity of the biofilm that was formed, right? And we can separate in 4 different groups, depends on the amount of biofilm formations. We also try to analyze for correlations between the biofilm forming abilities to another character, such as their violence for toxin productions, or maybe in the case of a classification system such as a rapprotyping. But unfortunately, we still do not see a significant correlations between biofilm forming abilities to another characters also, right? And after we predict that the endolysins might have shown a possibility to be a phage depolymerases enzyme. So I just kick-started with subclone, express, and purify to get a recombinant endolysins. And after that, I challenge it to the difficile biofilm. And the results turned that it's quite being likely a dose-dependent manner that this enzyme can degrade biofilm by noticeable fragmentations of the biofilm into smaller pieces also. We also try to use a crystal wireless staining assay to tracking on the quantity of the biofilm, but unfortunately, we still have a challenge about this part also, because comparing to another bacterial species to the difficile, this one is quite from a very fragile biofilm comparing to other. And so to solve this problem, we also use another analysis that is the mechanical analysis to look into the topologies and the intricacy of the biofilm by analyzing for Young's value also. And the results turn that comparing between the biofilm that treat with the enzyme that or the control, we can see a decreasing around 17.54 of the intricacy of biofilm when we treat it with the endolysin. right? And into the conclusions, right? That's my works that emphasize that the formations of the difficile biofilm exhibit a variability across the different stains as several studies suggest. And also, our phage isolates also can successfully inspect and kill the cell inside the biofilm populations, even though they are under the matrix of the biofilm. And the biofilm that was treated with our enzyme can reveal significant degradations by noticeable fragmentations of the biofilm. And last but not least, that is the alterations in the pathologies and the intricacy of the biofilm might be one of the evidence that our enzyme can do something with the biofilm state of the cell, right? And for our further study, we also plan to extract just only the matrix of the biofilm, right? Because of the biofilm populations, they compose of two major components. The first one is the cell, the second one is the matrix. So we want to just only extract for the matrix, and after then we will separate into different groups, the control and the treatment group with the enzyme, right? And after that, we will separate each component by using SDS page, and we might see a noticeable changes in some band of the components. For example, we might see a increasing of the byproduct from the depolymerases and maybe decreasing of the substrate of the enzyme also. And that specific components might be analyzed for further by using the mass spectrometry, right? And we also plan to do a Raman microspectroscopies also, because some enzyme might be not have effectively degrade in the biofilms formations, might be slightly affect. So we want to know that does our enzyme have the effects or not by using the Raman spectroscopy to tracking on the distributions of the chemical compound inside the biofilm, right? By reflecting in the Raman spectra and also the Raman map also. And my work cannot be successes with the meaningful guidance and helpful from my team from the Department of Biochemistry, our team from the Department of Biology, and also our team from the School of Material Science and Innovations also. And I might have not this invaluable experience to miss all of you here if without the funding from the Faculty of Graduate Studies by Mahidol University also, right? And this is the end, and this is my Q&A session. Happily to meeting all of you here. This is my first international conferences also. And if you have any questions, I'm pleasure to answer all of them.